Namaskar, welcome to PK's classes. Today we are going to discuss about the two RNA which are involved in RNA silencing or RNA interference. One is microRNA and other is siRNA or small interfering RNA, and what is the difference between these two. As we know, in RNA silencing on or RNA interference, we use the non-coding RNAs like miRNA and siRNA. As we know, the coding RNA is messenger RNA, which has the genetic codes for the protein synthesis, and the non-coding RNAs can be the housekeeping RNA like tRNA and ribosomal RNA. And these are the regulatory non-coding RNAs, microRNA and siRNA. And before discussing the difference between these two, let us discuss how they function. So microRNA is produced from the microRNA gene by the action of RNA polymerase II. First there will be production of the precursor of the microRNA, that is the primary microRNA consisting of about 1000 nucleotides. Then by the action of RNase 3 family of enzymes like DROSA in addition to DGCR8 in mammals, PASA in Drosophila, there will be about 70 base pair pre-microRNA, and then this pre-microRNA through the nuclear pores from the nucleus enters into the cytoplasm by the transporter exportin 5. Pre-microRNA in the cytoplasm it will be the substrate for the enzyme dicer, and this dicer now forms the miRNA duplex, about 22 base pairs. Now in the cytoplasm there is RNA induced silencing complex RISC and argonaute protein. So it is not specific to any argonaute protein. Any argonaute protein can bind. So this is the argonaute protein and this is the RISC complex. And what will happen? This RISC complex will separate the sense strand, and the sense strand will be removed from this duplex of microRNA, and the antisense strand will be there, and this antisense strand of the microRNA now binds to the target messenger RNA, and there can be either extensive match or less extensive match, that is fully complementary or partially complementary. And in case of microRNA there is partial or incomplete base pairing, and mostly, and that causes incomplete base pairing, that causes so let us write incomplete base pairing, that will cause the formation of P body, and there will be ribosomal block and it will repress translation. However, if there will be extensive match or complete base pairing, then that will release the RISC RNA induced silencing complex, and there will be rapid mRNA degradation. However, mostly in case of microRNA this incomplete base pairing occurs. Now siRNA or small interfering RNA is exogenous. Say suppose there is virus infection, then that virus will enter into the cell and release the double-stranded RNA, and that double-stranded RNA again by the action of dicer like microRNA in the cytoplasm will break the double-stranded RNA into small interfering RNAs of about 21 nucleotides. Then in the cytoplasm, again here the RISC, the RNA-induced silencing complex, and basically argonaute 2 AGO2 protein will bind the form the complex. So this is the RISC complex, the siRNA, and this is the argonaute 2. And this complex will remove the sense strand or anti-guide strand, and the guide strand or antisense strand will remain with RISC, which will bind to the target messenger RNA, and again, like in microRNA, there will be either complete base pairing or incomplete base pairing, but mostly there is complete base pairing causing mRNA degradation. Incomplete base pairing causes ribosomal block and stop the translation. In addition to its action in the cytoplasm, it also enters into the nucleus, where it binds to the DNA. And forms another complex called as RITS. The full form is RNA induced transcriptional silencing. Here T stands for transcriptional silencing. So what it causes? It causes histone methylation, which leads to chromatin remodeling, which causes the heterochromatin formation. And once there is heterochromatin, there will be no transcription, because transcription occurs when it is the chromosome is in the form of euchromatin, and heterochromatin formation leads to no transcription or transcription repression. So now let us see what is the difference between microRNA and siRNA, or miRNA and siRNA. miRNA full form is microRNA, and siRNA full form is small interfering RNA. MicroRNA is endogenously expressed, siRNA is exogenously derived. As we have seen this, that microRNA is formed from the microRNA gene, whereas the source of siRNA is exogenous. Suppose a virus infection, it provides the double-stranded RNA from which siRNA is formed. So microRNA is endogenously expressed, siRNA is exogenously derived. Then microRNA is a heteroduplex and siRNA is a single duplex. MicroRNA is found in the plants and animals, including higher mammals, and siRNA is found in lower animals and plants. It is not found in the mammals. Then microRNA is usually 22 base pair. It can vary from 18 to 25 nucleotides, and siRNA is 21 base pair, and it can vary from 21 to 23 nucleotides. MicroRNA is capable of targeting over hundreds of messenger RNA types at the same time. It can target many messenger RNA, but siRNA is capable of targeting only one mRNA type. Prior to dicer processing, so microRNA in the cytoplasm, this dicer enzyme is there. So microRNA, prior to dicer processing, in its precursor microRNA form, it is available, containing 70 to 100 nucleotides with scattered mismatches. Whereas prior to dicer processing, it is a double-stranded RNA molecule containing 30 to 100 nucleotides. So you can see this dicer is a cytoplasmic enzyme, and siRNA is formed by the action of this dicer enzyme, whereas in microRNA, first it will form the pre-microRNA, and this pre-microRNA is the substrate for this dicer enzyme. And then Next one is there is only use of RISC or RNA-induced silencing complex in case of microRNA, but in siRNA there is both RISC and RITS, that is RNA-induced transcription silencing. As we have already discussed that Argonaute protein 2 is required in case of siRNA, whereas any protein from Argonaute family, not necessarily Argonaute 2 is required in case of microRNA. Then in case of microRNA, there is partially complementary base pairing. That's why there is basically ribosomal block and repression of translation. Whereas in case of siRNA, there is fully complementary to the target messenger RNA, complementary base pairing occurs. So that causes mRNA degradation. And there is also endonucleolytic cleavage. So we can say that there is in microRNA there is only repression of translation. In case of siRNA there is both repression of translation as well as transcription. So if we see their use, microRNA serves as a drug target, therapeutic agent, and diagnostic and biomarker tool, and siRNA is basically it serves as a therapeutic agent and used in gene therapy. And if you see in the market, there are 4 FDA approved drugs, siRNA, used for different diseases, different orphan diseases rather. And microRNA based drugs have not yet passed phase 3 clinical trials. So basically microRNA, we can say they have some physiological functions. They regulate development of cell, maturation of cell, differentiation of cell, apoptosis of cell, cell signaling, cellular interactions, homeostasis, all these things. Whereas the drugs like patisiran, givosiran, lumasiran, inclisiran, all these FDA approved drugs are there, and inclisiran is used to reduce LDL cholesterol in people with familial heterozygous hypercholesterolemia. Patisiran is used in patients with polyneuropathy and cardiomyopathy associated with hereditary amyloidosis. Givosiran is used in acute hepatic porphyria in adults. Lumasiran is used in hyperoxaluria, that is a condition in which oxalate is increased in urine. So siRNA is now available as drugs for different conditions, whereas miRNA is not yet still available for use as drugs. So this is all about the difference between microRNA and siRNA. Thank you.